Hi, I'm Elena Rossini and I'm doing bacterial gene regulation. What is bacterial gene regulation? It is, quote, the process of turning genes on and off is known as gene regulation. Gene regulation is an important part of normal development. Genes are turned on and off in different patterns during development to make a brain cell look and act different from a liver cell or a muscle cell, for example, end quote. It is a regulatory mechanism that the RNA can control the transcription of only certain numbers of genes, or it can control many genes all at the same time. Some key points. The sigma factor is the subunit of the RNA, which recognizes specific promoters that are needed for growth conditions. Another key point is that there are DNA binding proteins, which can be a repressor, which is a protein that prevents transcription, or an activator, a protein that enhances transcription. This is an example. It's lac operon. This operon codes proteins that are involved with the transportation and hydrolysis of lactase. Is only turned on when lactose is present and not glucose. When glucose is present, it prevents the operation of lac operon genes, which makes sure that the cell uses the glucose before they use up the lactose. And this on the bottom right is just a diagram of bacterial gene regulation. Thank you.
Okay, so DNA replication is a basic fundamental process in a cell. It creates two daughter cells with the exact same DNA from one DNA, and each strand of the existing DNA is a template for replication of a new DNA. The purpose of DNA replication is accurate duplication of genetic material in microorganisms. It's needed for transmission of genetic information from the parent to the offspring. It allows microorganisms to grow and adapt to changing environments, and it helps maintain genetic stability and diversity in a population. How it works in prokaryotes, DNA unwinds at the origin of replication, and then helicase opens it to form replication forks, and then binding proteins coat the DNA to prevent rewinding, and topoisomerase binds to prevent supercoiling. And then primase synthesizes RNA primers, and DNA polymerase adds nucleotides to the end of the primer, and then strands begin elongation, and RNA primers are taken out, and DNA ligase seals the gap between the two. So when they're like splitting, DNA ligase seals it so that there's one daughter cell in the other one. In eukaryotes, the difference is they use multiple replication forks, and in prokaryotes, they're just from one origin. And eukaryotes use more polymerases. So yeah.
Hello everyone, so my name is Logan and I will be doing horizontal gene transfer, specifically transformation. So horizontal gene transfer is a gene transfer between two already existing organisms. The donor transfers genetic material to a recipient organism that is not the offspring. So opposite of that, the vertical gene transfer occurs when parents pass genetics to the offspring. So transformation can take place between bacteria, plants, and non-animal eukaryotic cells, such as fungi. It is also beneficial to the recipient as it allows the bacteria to produce new proteins, which leads to new functionality and can allow for selective advantages for antibiotic resistance. Transformation typically takes place between bacteria of the same or closely related species and occurs when bacteria takes up free extracellular DNA and incorporates it into their genomes. Usually when one bacteria lyses or splits open, it releases its genetic contents, usually in the form of a free-floating plasmid, and can be integrated into the chromosome or exist as a plasmid. Then another bacteria comes and acquires it. How well a bacteria can acquire DNA is known as its competence. Thank you.
I'm Alexandra, and I have the second part of horizontal gene transfer, or transduction. What is it? Transduction is a type of gene transfer that occurs when a virus transfers DNA from one bacterium to another. Basically, the new bacterium acquires new genetic material and information. How it works. The donor cell will replicate its own DNA or genetic material, and then it will create a tube or pathway that connects itself to another cell. This tube is known as a sex pilus. The DNA travels through the pilus as a single linear strand into the new cell. There are two different types of gene transfer through transduction. These include specialized transduction and generalized transduction. The first type, or specialized transduction, the virus packages only genes on or near the attachment site, and only a select few of these genes are transferred into the next new cell. But in the second type, generalized transduction, any bacterial cell can be packaged and transported into the new cell.
I'm Alana Sutton, and I will be going over horizontal gene transfer, the conjugation part. So the key points of conjugation, it's a direct cell-to-cell contact. Requirement is conjugation requires direct contact between two bacterial cells. Pilus formation. A donor cell extends a pilus to attach to a recipient cell. Transfer of genetic material. Plasmid transfer. A plasmid, a small circular DNA molecule, is often transferred from the donor to the recipient. F factor. In many cases, the donor cell contains a fertility factor plasmid, which enables the formation of the pilus. Steps of conjugation. Attachment. The donor cell attaches to the recipient cell via pilus. Connection. The pilus retracts, pulling the two cells closer and forming a bridge-like connection. DNA transfer. A copy of the plasmid DNA is transferred through the bridge from the donor to the recipient. Separation. After the transfer is complete, the cells separate and the recipient cell now contains the new genetic material. Results. Genetic variation. The recipient cell gains new genes which can confer advantages like antibiotic resistance or additional metabolic capabilities. Why it's important. Genetic diversity. Increased variation. Conjugation promotes genetic diversity in bacterial populations, allowing them to adapt to changing environments. Antibiotic resistance. Resistance genes. Conjugation can spread antibiotic resistance genes among bacteria, making infections harder to treat. Public health impact. Understanding this process helps in developing strategies to combat antibiotic resistance bacteria.
Hi, my name is Grace, and I'm going to be talking about plasmids and transposons. Plasmids and transposons are two different types of genetic elements that have the potential to play a crucial role in gene transfer and microbial evolution, particularly in bacteria, but also in other microbes. Let's start off with plasmids. Plasmids are extrachromosomal DNA molecules that are much smaller in size than a chromosome. While plasmids are not essential to the life of bacteria and the normal function, they do have a huge advantage in bacterial life. First, let's talk about the structure and composition. One standout characteristic of plasmids is that they replicate independently. This means that they have their own origin of replication. While plasmids do not carry essential genetic information, they do carry genes that code for selective advantages. The most common way that plasmids transfer these advantages is through conjugation. The plasmids that transmit through cell-to-cell contact are called conjugative plasmids. Some plasmids are also considered non-conjugative, meaning that they can only be transferred with the help of conjugative plasmids. Episomes are also a subset of plasmids that are able to integrate themselves into the chromosome of the cell, which allows their replication to then be regulated by the chromosome of that host bacterial cell. Now we move on to function. There are 5 main types of plasmids that all have different functions that provide a specific advantage to their inhabited microbe. First, there's fertility plasmids. These plasmids contain genes that allow for conjugation from one bacterium to another. There are also episomes, and a bacterium would be considered an F-positive bacterium if it contains an F plasmid, and an F-negative bacterium if it doesn't. The next type is resistance plasmids. These plasmids contain genes that allow bacteria to better defend against antibiotics and environmental poisons. When these plasmids conjugate with bacteria, they form drug-resistant strains of bacteria. Next, we have virulence plasmids. These plasmids turn their host bacteria into pathogens, which allows them to quickly spread and replicate. E. coli and Salmonella are examples of bacteria that contain many virulent plasmids. The fourth is degradative plasmids. These plasmids contain enzymes that aid their host bacterium in breaking down and digesting compounds such as salicylic acid, also known as aspirin. And the final is coplasmids. These plasmids code for bacteriosomes. These are proteins that attack other bacteria and make space for the host bacteria to grow. Now we move on to transposons. Transposons, also known as jumping genes, are a sequence of genes that can change position from one part of the genome to the other. There are different classes. There are transposons class 1, which are retrotransposons, and class 2. Class 1 transposons, also known as retrotransposons, are classified as DNA that has to first become RNA and then use the enzyme of reverse transcriptase to make a copy and move to a new location on the genome. Then we have class 2 transposons, which contain DNA that moves from one place on the genome to the other without the use of that enzyme. Transposons are also considered mutagens, meaning they cause mutations of the cell. Insertion of transposons can cause damage to cells, change, or even destroy gene activity. Now, why are they important? Transposons are important in creating genetic diversity and reconstructing the genome. Through shuffling the genes, they enable bacteria to evolve their immune systems and disease processes. Thank you.
Hi, my name is Caitlin Faust, and I will be talking about mutant selection. Basically, this is where a cell has the rare occurrence of mutagens that can cause the cell to spread mutant cells and shift a parent cell. This can lead to a disease resistant to medicine and can cause doctors to be unable to assist patients. There's two categories, which is direct selection and indirect selection. In direct selection, this is when the original cells are not able to adapt, but the mutant cells are. They begin to grow in places that the original cell before division was not able to grow in. Isolating these cells is much easier than indirect selection. More and more of the mutant cells are being produced while the original is not producing as much of itself. An example of this is tuberculosis, which is becoming more and more resistant to antibiotics, leading them to having no effect on the disease. The cells are now mutant and stronger. And then the other category is indirect selection. This is a way of finding the autotrophic mutant back to the parent cell. They use replica plating, where a master plate containing autotrophic and prototrophs are spread on the agar plate and set until both colonies separate. They then transfer the plates onto a velvet cloth, and then a glucose salts agar is able to pick up the prototrophs, but the autotrophic can only grow on the nutrient agar plate and not the glucose salts agar. The scientist is now able to see which colony is which because the fact that one can't grow on a certain agar plate and one can. One of the possibilities is cancer, so screening for possible carcinogens is very important because mutagens are forms of carcinogens which are cancer causing. One test they do is the Ames test, which is a method to test the mutant on a bacterial strand to see the effect it has on growth. This can show if the colony duplicates it or matches the control plate by not growing. This can allow patients to know whether they are dealing with cancer or just a mutant.
In the Christian worldview, healthcare personnel are called to be good stewards of human health, reflecting God's love and compassion towards those who are suffering. 1 Peter 4:10 says that each of you should use whatever gift you have received to serve others, as faithful stewards of God's grace in its virtuous forms. This verse emphasizes believers to actively engage in serving others using their God-given gifts. It suggests that serving is not just an obligation, but a way to express gratitude for the grace they have received. By doing this, they reflect God's character and contribute to the well-being of their community. It also highlights that the interconnectedness of believers is in the body of Christ, and everyone plays a vital role in supporting one another and fulfilling God's purpose.
Healthcare personnel are encouraged to embody compassion and empathy, mirroring Jesus' healing ministry, just like in Matthew 14:14, where he healed the sick out of compassion. A Christian perspective promotes holistic care that addresses not only physical ailments, but also emotion and spiritual needs, recognizing the inherent dignity of each person created in God's image, as said in Genesis 1:27. Just as Jesus advocated for the marginalized, healthcare workers are called to advocate for those who are suffering, ensuring that everyone has access to the care they need. Caring for the suffering is seen as a normal obligation and a reflection of one's faith. This aligns with Matthew 25:40, where serving the last of these is equated with serving Christ himself. Overall, by embracing the role of good stewards, healthcare personnel not only fulfill their professional duties, but also live out of their faith, contributing to the healing and well-being of individuals and their communities.